Welcome everyone to the latest talk in Fishes series on evolution in finite populations. Today's speaker will be Srishti Patil. She was a master's student at ICER Pune. She's now working at the German Cancer Research Center, and today she's going to be presenting to us work she had done during her master's thesis, working with Dr. Mark Noble on preventing evolutionary rescue in cancer using a two-strike therapy mechanism. So, take it away.
Wait, did you say Mark Noble?
What was the name? Is it?
Rob Noble, yeah.
Oh, it's Rob Noble.
Yeah, it's like both very generic names. It's fine.
Yeah, I think Mark Noble's a football player for West
Uh,
Ham.
mhm. Okay, so I'll start.
Yeah.
Um, yeah, so I'm Srishti, and the topic of my master's thesis was preventing evolutionary rescue in cancer using two-strike therapy. Right now, I'm not doing work related to this evolutionary rescue or I'm working on colorectal cancer evolution, and that's more about how tumors originate and modeling that, and it's more data centric. Um, but this project was where I started working sort of at the intersection of theory and application. So this kind of work is where you kind of need to, in addition to the robustness of your results, you need to demonstrate the practicality of it. So a lot of times models are quite far from application, but like in this kind of work, you need to justify why you're even doing it. So evolutionary rescue, I will define that first. It's a process by which a population that would have gone extinct in the absence of evolution, it persists due to natural selection on heritable traits. Um, and so if extinction doesn't happen, then it means that evolutionary rescue has occurred, and that's basically relapse, and we want to prevent relapse. And specifically, the question is, suppose a cancer treatment fails because a small fraction of tumor cells are resistant. Would we still be able to cure the patient by switching to another treatment at just the right time? So, we want to find the most effective way to implement two cancer treatments sequentially. And the sequentially part is important because this work doesn't compare this type of treatment to, say, combination therapy, where you apply multiple treatments at the same time. Um, so this is limited to sequential treatment, and we don't know if it's better or worse than combination treatment. That's one of the major questions. Um, so we want to see if somehow applying treatment sequentially can help us exploit the vulnerability of this resistant population when it is small, because any treatment that works would make the population smaller. And if there is some point where we can use this sequential treatment to take advantage of that vulnerability, that's what we want to model. And I'm talking about all of these things in very abstract terms right now, um, because I want to start sort of outline how you would build a model um, about this or this type of model, and that way it would also be easier to understand what assumptions we're making and why we're making these assumptions. So to model the system, we have to think about three main phases. One is, of course, before treatment, and one is during each of these treatments. And we are talking about two-strike treatment, two-strike therapy. So it's two treatments, one after the other. And we are trying to build the simplest model possible that would model the vulnerability. And this is because we are not trying to fit any data because we don't have any data. And we want to keep the model simple because we want to interpret it. There's another argument that models should be as complex as possible to capture the, I don't know, reality. But that's more important when you have an experimental support to actually verify it. But right now, we want to keep the model simple. And that motivates our first assumption, that is that during treatment one, there is no chance of extinction. So this, I think, is a reasonable assumption because we would need to use a two-strike treatment only if the first one doesn't work. So we assume that there is no extinction during treatment one, and this makes our lives very much easier because we only have to consider relapse after the second treatment, and we have to give both treatments one and treatment two. So in the before treatment phase, we need to think about how much diversity exists, how much resistance already exists, and what is the size limits on how this treatment, or this limit under which this treatment would be useful. Treatment one and treatment two phases, we have to think about how the population would react to the treatment, and how the resistant population would react to the treatment, and how these subpopulations would interact with each other. So the simplest model is an OD model that describes this population dynamics. And because we have two treatments, we have four cell types. The S cell type, that is sensitive to both the treatments. The R1 and R2 cell types. So R1 is resistant to treatment 1 but sensitive to treatment 2. And R2 is sensitive to treatment 1 but resistant to treatment 2. And R12 is resistant to both. That's a pretty standard model setup for two treatments. And we have mutation rates to go from one cell type to the other. And if you see, the mutation rate from S to R2 is the same as R1 to R2. So it's sort of like the rate of acquiring resistant to a treatment. So acquiring resistant to treatment 2 occurs at rate mu2. And then we have the dynamics. We have logistic growth. And here GS is the growth rate of S cells, and delta 1 is the treatment level during, like the treatment dose or a measure of the treatment dose. Mu1, mu2 is the mutation rates. And so here we have the growth terms, here we have the mutation terms. And with the initial conditions, we can model this. So what happens on a broad scale during treatment one is that the S population would decrease because it's susceptible. R1 population would increase because it's resistant to treatment one. R2 population would slightly increase and then decrease. This is because we have a lot of mutations from S to R2 cells because there are a lot of S cells in the beginning of the treatment. So of course, this depends on the initial conditions, how many S cells and R2 cells we have in the beginning. And it also depends on the mutation rate, how fast this mutation is occurring. But in realistic values of mutation rates, we see that there's a slight increase in the beginning and then a decrease. And then the R12 population size increases because it will increase throughout the treatment because it's resistant to both the treatments. Then we switch to treatment 2, and this is a schematic of like the total population size during the two treatments. And here we have these dynamics, and then the population could either go extinct after switching to treatment 2, or if you don't switch to treatment 2, this is what would happen. The R1 population would increase. And if you switch to treatment 2, there could also be evolutionary rescue, but that is the evolutionary rescue that we are trying to prevent. So what happens during treatment 2 is, as population decreases, it's sensitive. The R1 population also decreases because it's sensitive in the second treatment. And the R2 population and the R12 population, they would increase. And that is why we call them potential rescue mutants, because these are the only cells that can lead to evolutionary rescue. Now, this connects to the assumption that we made earlier that there is no evolutionary rescue in treatment 1, because now we can only focus on these two cell types and we don't have to think about the R1 population, rescue due to R1 population, because we know that, uh, yeah, the assumption. Um, so even if a single R2 or R12 lineage escapes stochastic extinction over the course of the treatment, then we would have rescue. So this is what we want to model. Um, what is the probability of these lineages going extinct and not escaping stochastic extinction, because they would be very small at some point, and if at that point they go extinct because of stochasticity, then that is the only condition under which there would be no evolutionary rescue. um, Okay, so now we want to track the lineages of R12 and R2 cells. And it boils down to a very common question in this field, which is that of emergence and establishment of rare traits. Because these two cell types are the only cell types that have a positive fitness in treatment 2. They um, they will start from a very small population. This is a very commonly modeled system, you can say. But the difference here is that we are in this two strike system, and we want to take into account what happened during treatment 1 as well. So the goal is to maximize the probability of extinction of R12 and R2 lineages. And to do that, we then go to evolutionary rescue theory, but I will explain what all of this is. Um, so intuitively, we want to minimize R12 and R2 cells, like the number of cells. But we have to look at the whole duration of the treatment, because they will come up throughout the treatment. Like the resistance is being acquired throughout the treatment. And now we need to make an assumption of how this resistance is being acquired. So we need to sort of see over time how these mutants are being generated, the potential rescue mutants. And for that, we make an assumption that the generation of rescue mutants with time is a Poisson process. And because it's a Poisson process, the probability of extinction would mean the probability of um, mm, like in the, oh shit, I forgot to include a Poisson formula here. But if you guys know, the probability of zero number of lineages would be e to the power of minus times the rate of generation of these mutants. So this is the exponential and the negative of the rate of generation of these mutants. And so now we only have to find the rate of generation of these mutants throughout the entire treatment. And this is what we do here in this formula. So I will start with rescue mutants that are generated after we switch to treatment 2. And they are called de novo mutants. So that's where the DN comes from. And these are mutants that are resistant to treatment 2, and R12 and R2, like we said before. And so this is the rate of their generation. So this is number of S cells times mu2, number of R1 cells times mu2, because these are the cells that can acquire resistance and give rise to new R12 and R2 lineages. So if S acquires the resistance and S cell acquires the resistance, then you would get R2 lineage starting. And if an R1 cells acquires resistance, then you would get an R12 lineage that begins. And pi e is the probability of extinction of that lineage. So if a lineage begins, there is a certain probability that it would escape stochastic extinction. And I mean, that is a very important part of the formula, and we will come to it later, like we will derive it later. But uh, right now, this is the probability of extinction of that lineage. I'm sorry, the probability of establishment, the probability of escaping extinction.
So,
And
Hey, there seems to
Yeah.
be a question in the chat. Let me know if you
Oh
want to
yeah, I
like
can't see the chat.
later.
Sorry.
Uh,
Yeah, yeah,
I'll
yeah.
I'll I'll feed them to you, but like, just let me know
Yeah,
if you
yeah.
want to take them at the end or if you want to take them as we go.
No, 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 no. Yeah, please, as you have any questions, please ask. Yeah.
All right. So the first question seems to be about. He's asking: is there a specific reason why a Poisson process is a good model for this situation, or is it just an arbitrary choice? So maybe if you could motivate that, that would be very helpful.
I mean, it's a common choice because it's like, I mean, it's used to model any process that has a constant rate over time. Like if you have a rate of something that happens certain times over a minute, like certain times a minute, then you could take that rate and model it in a Poisson way. That is like it's um yeah, you of course assume that all of these events are independent. So if you take like make all of these like very easy assumptions, then you get the simplest is the Poisson process.
I have a question about the first slide you had some logistic equations. But those were deterministic. I kind of lost
Mhm.
uh, how you come to these equations. I thought they would capture the dynamics and then you would tinker with them. But yeah, there you have stochastic dynamics. I
Yeah, yeah, yeah. So the OD model, this one, is used to model the dynamics of bigger populations because it's deterministic. And when we talk about the rescue mutants, which is the R12 and, oh, sorry, the R12 and R2 cells, we need to model them stochastically. So in here, for example, this S term, the dynamics of that we would get from the OD model, the S R1 here. And this one, the number of R2 cells at the time of switching, which we call tau, that we would get from the OD. So input to these equations we would get from the OD, the solution of the OD. And this is only to model the R12 and R2 cell lineages or their extinction.
okay. So am I like correct in thinking that it's sort of modeling at like a smaller time scale how processes occur? It's,
Smaller size scale,
okay.
because this is like this. So these formulas, they give you the probability of extinction of these lineages over the entire course of the treatment, because we are basically integrating over time, from 0 to tau and then tau to infinity. So infinity time, do any of these lineages ever uh, come up? So the
Okay. Okay.
probability of extinction would be that they never come up, or
Okay.
like
Okay.
if they come up, then all of them go extinct. Yeah.
Yeah, yeah. I now get it. What it is. Yeah. Thanks.
Hmm.
Thanks.
Okay. Yeah, so this is I haven't explained this ever to anyone before, so if something is unclear, please ask questions. Okay. So again, so here I was talking about the de novo mutants, which is mutants that come up after switching to the second treatment. And they are generated when S cells acquire resistant and turn into R2 cells, and R1 cells acquire resistance and turn into R12 cells. So the rate of that happening is S times mu2 and R1 times mu2 integrated over tau to infinity, because we are talking about the second treatment. And pi e is the probability of establishment of these lineages. So the probability of these lineages that we counted here, of them not going extinct. Yeah, I always get confused at this part. So this entire expression is the probability of uh, there being no rescue lineages that escape stochastic extinction. Is that clear? If there are any questions about this, the probability of the PDN, then let me know.
Hello? Hello? Can you hear me?
Yeah, 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 yeah,
Yeah, hi. I had a question regarding this pi e term.
yeah.
So, are you saying this is like related to demographic stochasticity? Like it tells you the probability of
Yeah,
extinction for a given population?
yes, yeah.
So, like, won't it depend on your population size of that lineage?
So that is another assumption that I will come to later. We assume density independence when these lineages come up, and we justify that assumption. Why we can assume this generation of rescue mutants to be density independent.
So, are you treating pi as a constant?
No, I mean it's constant for a certain set of parameters, but yeah, so it doesn't change with time.
Okay, but and it does not change with the uh, population of that lineage?
No, it does not
Population
change with
size,
the
I mean.
population. Yeah, no, it doesn't.
Okay,
So
I
I
have to
will
think about it.
come
I'm a
because
bit confused. Yeah.
yeah, so that's because we are assuming density independence that we can not take into account the population size.
Okay, sure. Thank you.
Yeah, I will come to that later. So, so this is the probability of the newly generated, which is in treatment two, those newly generated lineages, all of them going extinct. And this one, the PSGV, is the probability of mutants that existed throughout or that existed at the time of switching. So they were generated throughout the treatment one, the probability of those going extinct. So they are again, the probability of R1 acquiring uh, resistant to treatment 2, so mu2, or R2 acquiring resistant to treatment 1, which is mu1. So this term counts the generation of R12 mutants in the first treatment. And this term is, we have taken the R2 population from the ODs, and that we have assumed to be Poisson distributed with the expected value of R2 to be the value from the ODs. So we solve the ODs during treatment one, we get a value of R2. We take that as the expected number of R2 cells at that time, and we assume that it's distributed in a Poisson distribution. And from that, we again get the probability of them being zero, which is e to the power minus the expected value. And that is exactly this part of the equation. So this is an assumption, again, that this R2 population is Poisson distributed. But um, we have done a lot of work to see where this assumption is valid and where it's not. And for most reasonable or parameter values of interest, it's valid. And this assumption is again very useful because then we can find out the probability of extinction of the R2 mutants from the first treatment. And we don't need to model the growth, like stochastically, we don't need to model the growth. So this is the probability of extinction of potential rescue lineages that came up during treatment one. And this is the probability of extinction of potential rescue lineages that came up during treatment two. And we want all of them to go extinct. So we multiply both of these terms, and then we get the total probability of extinction given the time of switching. So this comes up here, 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 here. So we are essentially finding out or maximizing the probability of extinction over time. And by varying the switching time, the time at which we switch from treatment one to treatment two. Okay, any questions at this point? Okay, So then we can talk about the pi e term, which is the probability of establishment of a rescue lineage. So here, there are many ways to model this. This is a very commonly used expression. So if we assume a simple birth death process, we would get this formula for the pi e, but this makes an assumption of the birth death process. If we want a more general form of this expression, we can use a continuous time real valued branching process and uh, use its generating function and then find out the pi e that way. We tried both of these options or whatever, and the results are pretty much the same. So it comes down to the assumptions, which is that this pi e term is density independent. And this is an assumption in a lot of such models where you model this emergence. And we show that the values with competition of pi e, which means that if pi e depends on the population size, then the value of pi e, it converges to the value without competition over time. So after some time, it would be the same, or pretty close, with or without competition. So density independence is a reasonable assumption because this initial time is uh, uh, what do you say? I mean, yeah, because this initial time is small, very small compared to the time scales we're looking at, like the time scales of treatment. Okay. So now we have the establishment probability, and using this, so this depends on the growth parameters. And using this, we can solve these equations, and we can get the probability of extinction of the entire population. So to solve these can be solved explicitly, but you have to make some more assumptions about density independence. If you don't make those assumptions, then we solve them numerically, uh, and that's the results that I will show, the ones that we solve numerically. And in addition to this whole approach that I just explained, we also have stochastic simulations, which is just a Gillespie algorithm to see the dynamics of the population over time when we apply these treatments. So in the simulations, we just have a bunch of cells. We apply the first treatment, which would change the death rate of these cells. And then we would switch the treatment at some point, and then we would count the number of times this goes extinct and the number of times it doesn't go extinct. And that's how we get the probability of extinction from the simulations. And then we can compare our predictions, analytical predictions, to these results. And here you can see more like to get a better intuition of what would happen. So for example, if we have a small switching size, so again, we can talk about switching times, or we can talk about switching sizes. And here we chose to talk about switching sizes. We could also talk about switching times, but it's the same thing. And it's an important point because sometimes the results would or the interpretation would depend on which kind of implementation you are using for the treatment. So it depends on what you are using as a cue to switch the treatment. If you're using switching size as a cue, so if the population reaches this size, then I would switch. That is a population size cue. Um, then it could be a different interpretation when compared to I would switch after a certain amount of time. but that's like coming to the more clinical side of things. But here it's equivalent if we talk about switching time or switching size. If we have a small switching size, then you see that a lot more instances of this simulation would go extinct compared to when we have a large switching size, because the population is more vulnerable here in the small switching size case. Okay, so this is where I stop talking about the model and I'll talk more about the results. So are there any questions?
I have a couple actually. So,
Mm.
so you say you use the Gillespie algorithm, right? So, what were the population sizes you were dealing with, and are those realistic to what you would see in an actual setup? Like, I'm not very familiar with cancer
Yeah,
research, but one thing which was
yeah,
very
yeah, yeah.
annoying with the Gillespie for us was 10 to the 5, anything bigger than that, and it really takes a very long time to simulate. So, what?
Yeah, yeah. So 10 to the 6, that was our size, and it's not realistic at all. No, like a 1 cubic centimeter tumor would have a billion cells. So
Oh no.
that's not realistic. But and that is actually one of the biggest practical considerations when we talk about these treatments, because this is well below the detection limit
Yeah.
of what clinics can do. And so it's only a prediction of what would happen on a bigger size. We can make educated predictions, of course, that if 2 strikes don't work, then in principle 3 strikes should go even further, and you would be able to use bigger tumors, uh, not use, but like, cure bigger tumors. Um, so yeah, so this is at, like, this size is not realistic, but in the paper also we have discussed this and we've talked about what could be done for bigger tumor sizes, or uh, how this hints at this therapy still being useful for bigger tumor sizes.
I see. The other thing was you seem to be assuming that these replicate as if they are asexual organisms almost, right? Like either they copy
Hmm.
themselves or they mutate.
Yeah,
Is that common for tumors, or like is that
yeah.
something tumors do all the time, or would you have preferred to do a different population genetics, but this is simpler, so that this is what you've done?
I mean, modeling it as like, yeah, an asexual organism, that is, they are pretty common. That's what most models on cancer growth look like, or cancer growth or cancer dynamics. And it is also simple. So yeah, that was it's like conventional, I would say.
No, no, I understand. I understand it might be conventional.
Mm.
I'm saying since you've also done more work after on
Mm-hmm.
cancer, is this a
Yeah, yeah, yeah.
realistic assumption? That's the like I would really like to know. I really don't know.
Yeah, so this is realistic in the sense that if you use such a model or this level of complexity in a model, and you have a specific aim. So, for example, if I want to model tumor growth to capture how do mutations accumulate when the tumor grows, then that this is a very specific goal, and this can be measured in experiment. So I have data that this is the number of mutations at some point, and then if we fit the data, then we get a reasonable fit. So we can get like, in that way it's a good choice of model.
Okay, thank you very much.
Yeah, of course. um so yeah, if there are no more questions about the model, um I will move on to the results. So again, I would emphasize that the stochastic simulations are independent. They don't assume any like evolutionary rescue theory or any of these Poisson distribution, anything. We are just counting how many replicates of the simulation go extinct and how many don't. Okay, so the main result or the main point of the paper, I think, is that optimal switching size is close to the population nadir, which is kind of obvious. And in general, it is better to implement the second strike after the nadir than before. So the population size being the smallest possible is intuitive, because that's when the resistant population would be most vulnerable, smallest. So the optimal switching size is close to that population minimum, which we call n min here. I hope it's not too small. And here we show the switching size the extinction probability against the switching size. And we see that if the switching size is lower, we have a higher extinction probability. And this red line is the n min, or the nadir. So we would have switching points that are before the nadir, before n min, which are shown in black, and switching sizes that came after n min, which are denoted by blue dots. So instead of taking switching time, or instead of using tau as our variable, we are using n tau. And because n tau is coming up twice, once before and once after, we are looking at before n min and after n min points separately. And this is useful because we can see that switching after the n min at the same switching size is generally better, as opposed to switching before the n min at the same size. Now this is because of the R2 population in the beginning. So remember when I said that the R2 population would increase slightly and then decrease? So that, and also the initial R2 population that is present in the tumor at the beginning of the treatment, that it takes time to decay, or it takes time to grow smaller during treatment 1. And because it's only resistant to treatment 1 and not resistant to treatment 2, it would only decrease during treatment 1, and you kind of want it to become as small as possible. So if you switch too early, then there is enough of the R2 cells remaining in the population that it's easier to escape, or there's more chance of escaping stochastic extinction. So that's why that is the source of this difference that you see. And to verify that, we look at the case of R20 being 0. And there again, you would see some difference maybe, but more or less it's the same. And this would again depend on accumulation of R2 cells, if that would happen depending on the parameter values. But again, like I said, for realistic parameter values, there would be some difference between the before nadir switching sizes and after nadir switching sizes. So this is an important, I would say, result. And here we show this for 2 treatment sizes. And yeah, so that is where we see that the optimal switching size is close, because the population extinction probability increases as we go closer to the n min. And we see that it's in general better to implement the second strike after than before. Now, we also had solving these equations with an assumption of density independence can give us explicit formulas for the n min and the optimal switching size. And there we found that the optimal point to minimize the probability of de novo rescue mutants going extinct is slightly after the nadir. It's always after the nadir. It's only exactly at the nadir if the cost of resistance is zero. So we assume here a cost of resistance because the growth rate of these two cell types, the S cells and the R1 cells, it's different because the resistant cells are assumed to grow slower than the S cells. Now, we also look at the case where there is no cost of resistance, but that is the only case when the optimum value for this term, the dn term, would be at the nadir and not after. So in general, this term is after the nadir, and this term is minimized. This term is minimized after the nadir, and this term can be minimized either before or after the nadir, depending on the parameter values. So it's not a robust result to say that the optimum is at or before or after the nadir, but because this term is almost always after the nadir, it's it's safer, so to say, to wait after the nadir to even find the optimal. And in general, the waiting after the nadir is better than switching before the nadir. So that was the main takeaway from this model. And we also look at a lot of other parameters, how this system changes or reacts to different changes in parameter values or conditions. And we found out that the optimum treatment level, uh, it also depends on um, which treatment you're looking at. So, okay, I'm not explaining very well. So I'll talk about the cost of resistance first. So we looked at the cost of resistance, and we see that it's not necessarily beneficial for treatment, and it depends on how the treatment is implemented. So when I was talking about switching times and switching sizes, so if we look at the extinction probability over switching size, then we would see that the no cost of resistance case, which is these darker colors, they are slightly better than the case with the cost of resistance. And that is counterintuitive because you would think that a higher cost of resistance, meaning a lower fitness for the resistant cells, would be better for treatment, but it's not if we look at this plot. But and it wouldn't be better if the treatment switches based on the population size. So at the same population size, you would get a better probability of extinction if there is no cost of resistance. But if the treatment switches in response to time, so switches after a certain time and not based on the population size, then this would not be true. So again, this is an important thing to consider, because when you design clinical trials on for a therapy like this, you would need to decide the trigger for switching to the second treatment. In some tumor types, the trigger would be more obviously the population size. For example, prostate cancer, where the population size or the volume of the tumor is proportional to this certain measurable chemical. And that's how you find out the size of the tumor. So in response to that chemical, you can then design a therapy protocol, a treatment protocol. But it's not so easy to do so in terms of time, because you don't know what the actual growth of the tumor is like. So this is why it's important to note which of these results are different when you change how the treatment is administered or implemented. So this was one of our major discussion points, because if no cost of resistance, which is the C equal to 0 case, is better than a cost of resistance, then it sort of uh, gives you a wider range of cases to work with. And this has also been an important point for adaptive therapy, which is another kind of say evolutionary therapy for cancer. And I will again discuss a little bit more about that. But the cost of resistance, let's say, has been an important discussion point in this field. Then we also looked at when two-strike therapy is feasible or for what tumor sizes, like we were discussing about tumor sizes earlier. And for the Gillespie algorithm, it was limited to 10 to the 6 cells. But for our analytical model, we looked at sizes up to 10 to the power 8. And we see that in those cases, it's not possible to achieve an extinction probability greater than 0.4. Now, let me explain this plot. This is plotting the probability of extinction, which is on the x-axis, and the population size, which is on the y-axis. But this is the population size below which it is possible to achieve, below which you will have extinction probabilities greater than Q. So N of NQ would be the probability, no, NQ is the population size below which you would always get the probability of extinction of 0.5. Is that clear? Of Q. Well, I don't know what I was saying. If someone doesn't understand this plot, please let me know. It's basically the population size versus extinction probability, but instead of plotting it population size versus extinction probability, it is sort of like an extinction probability threshold. So below this size of the population, you're guaranteed to get this much extinction probability. And we do this so that we can have a curve for different parameter values. It's easier to compare. But regardless, if this is not clear, then let me know. So yeah.
Seems like everyone gets it.
Yeah, okay. Because, oh my God, there was, for the peer review of this paper, all the 3 reviewers were confused about this plot. But yeah. So here we see that the dotted line, which represents initial population size of 10 to the power 8 cells, it ends here around 0.4-ish. So it's not possible to obtain higher extinction probabilities, even if you go lower in size. Which means like even if you, I mean, I guess it's not possible because you wouldn't be able to go very low. But anyway, in the two-strike case, you can't get an extinction probability of greater than 0.4. So our conclusion here was that it's only feasible to use two-strike therapy if our population sizes are uh, around 10 to the power 6, because then you can still get a good enough extinction, like pretty good extinction probability. And this also motivated a discussion on what could be like, can this be extended to bigger tumor sizes if we add more strikes? We also took a look at the carrying capacity to see if our assumption of density independence to solve the equations explicitly, how reasonable that is. And we see that the extinction probabilities are pretty much insensitive to carrying capacity. There is some change here, like if you increase the carrying capacity from the initial tumor size, which is N0, to 10 times that to infinity, there's not a lot of change. And especially after the nadir, if you're switching after the nadir. So our intuition or our interpretations from the explicit solving of the equations are reliable. Um, so yeah, so these are the results. These are the kind of results that we were looking at. We were looking at different parameter values of different variables and seeing how changing that can affect our treatment outcome. For the treatment level that I mentioned earlier, the treatment dose, or these terms in the equations, which so they would alter the birth and death the death rates. So if a cell type is under treatment, then it would increase the death rate of the cell. So that term is what we call the treatment efficacy. And for treatment efficacy, we mostly, like, the main takeaway is that higher treatments are better. Um, but we just found this very interesting little um, uh, thing, that if the treatment level of the first treatment is lower, um, then it is slightly better, but only if you switch before the treatment. So it was that if we are switching in one of these switching sizes, then it is slightly better to use a lower treatment dose. But I haven't shown this result because overall the optimum treatment efficacy values are, the higher the better, because switching after the nadir is better than switching before. So it doesn't matter which treatment level is better in the before nadir switching points, because the overall optimum is achieved switching after the nadir with as high treatment efficacy as possible.
I have a question about the
Yeah.
deltas. So like if you're trying to kill off a tumor, right? You can. I mean, I just want to ask why. You
Mhm.
use deltas over there. You could hypothetically reduce the GS instead, or the carrying capacity. Like, is there a reason why? If you want, you modify your logistic equations like that in
Mm.
when you're thinking. Yeah. So yeah, there are like three
It's
parameters you can tinker with. Yeah.
Yeah. No, I mean, it's because the delta terms are in like, per cell units.
Okay. Okay.
Um, I mean, it's
Okay.
like
Okay.
rate that depends on the population size.
Right, right. So I get it. So
Mhm.
like the therapy
Yeah.
targets is like a per cell, not really, population level. Like,
Yeah, yeah, yeah.
oh,
So any more questions about the results or the model? Because then I sort of go to the conclusion. Okay.
I think you can go to the conclusion.
Yeah. Um, so all of that, the results that we talked about and the type of model, it's to investigate if two-strike therapy should be used. And two-strike therapy, it can be used feasibly under certain conditions. That's what our conclusion was. And the biggest remaining question, I would say, would be to compare it to combination therapy, because that is an obvious question, that if you have two treatments, do you apply them together? Do you apply them sequentially? And there has been work on this, of course. But what I'm talking about is more under which conditions is it better to use two-strike therapy compared to combination therapy. Um, and in what conditions are both of them feasible, or in what conditions only one of them more feasible than the other. Then people also talk about long-term control strategies like adaptive therapy, which is like you adapt the treatment level according to the tumor size so that it doesn't grow out very fast. You have to sort of control the tumor size using the treatment level. So these during my work on two-strike therapy, I sort of understood this like general uh, conditions under which these three therapies could be useful, but there is no like we didn't go further on this. But this is like an open, I don't know, topic of research. Another um, possible next thing to do, or obvious next thing to do, is to see what happens if with the third strike. And that becomes, I mean, how many strikes can you give? It's you have a limitation on the number of treatments. Similarly, here in combination therapy, you have a limitation on the treatment dose because of treatment toxicity. Sometimes it's better to use one treatment in a higher dose compared to two treatments together in low doses. So all of those practical considerations are very important and the next step in this field of research. And I also wanted to talk a bit about the practical considerations of this model. And maybe like um, if this model, if we assume that this model is not correct, but if you want to apply this model to an actual clinic, like to actual patients, then what would be the main practical considerations? So the first one, or the yeah, I guess the main one is the tumor size and detection limits that we talked about. We showed that it's two-strike therapy is only possible for very low tumor sizes, and those are below the detection limits. But can we still design some treatment protocols that would take, use these takeaways from our two-strike therapy model to see which range of, I don't know, which range of time could be good to apply this kind of a switching strategy? That's one thing. The second is parameter values and what is realistic. This is something that I'm working more deeply on in my PhD project. So one of my aims is to understand how mutations accumulate with time. It's not straightforward at all to find out mutation rates, because mutation rates are like, first of all, how do you even detect that? It's not a well-known thing. So any parameter values that we use in the model, we don't know what are realistic um, ways to use these parameter values. And that is something to keep in mind for practical considerations, because then you need to work with parameters that um, can be detected, or parameter values that can actually be used from experimental data. And the third major thing is patient variability. All of these things are very, very different across all patients, across all tumor types, across all individual tumors. And designing any kind of practical strategy to use two-strike therapy would be this is one of the main things that needs to be done before you can apply any kind of treatment to a set of patients. Yeah, so that's it. Thank you for listening. This is